come back to our session. Uh, in this class, I'm going to explain you about absorption of drugs through GIT. Till now, what we discussed in the previous class, what is the basic concept of pharmacokinetics and biopharmaceutics in your by BPPK subject, right? You all understood? Okay. Then in that class, I have discussed about the passive diffusion of drugs also. In now, I would like to continue that session. So, in your syllabus, they mentioned under biopharmaceutics, okay, you have four topics. Those are absorption, distribution, metabolism, and excretion. From second unit onwards, you have pharmacokinetics topics. So under first unit only, you have four topics, right? So in that, first we discussed about biopharmaceutics and how the therapeutic index and how MEC and MTC will be happened. Okay, I explained briefly by showing the graphs and all. You got it? And next, I discussed pharmacokinetics importance. Then, what are biopharmaceutics? What is biopharmaceutics? The mechanisms which influenced your dosage form when you taken it orally, right? That is about biopharmaceutics. Under that, first we would like to discuss about absorption of drugs through GIT. So you all know about the structure of GIT and then how the process will be. In that, you need to learn about the mechanisms of absorption and factors affecting absorption of drugs. These two are there in your absorption, right? In that first point, the process of movement of unchanged drug from the site of administration to the systemic circulation is called absorption. So where you administer your drug from the site of administration to the systemic circulation is called absorption. The effectiveness of the drug can only be assessed by its concentration. So then how much concentration of the drug will be absorbed at the particular site? So it is very difficult to measure it. Instead of measuring the accurate concentration in plasma, there is a perfect correlation between drug plasma concentration and therapeutic response, right? You need not go directly to measure the concentration. How much concentration is enough to produce its action? So you have to attain always a perfect correlation between the therapeutic response as well as its drug concentration. That means at which concentration it is showing therapeutic effect, right? That is, the definition is again, if you take the drug from the site of administration, so you have to calculate the drug concentration from site of administration to the systemic absorption. That is nothing but, what do you mean by systemic circulation? Where your drug will be going absorbed or distributed, that is in plasma. That's why the definition came as process of movement of unchanged drug from site of administration to the site of measurement. That is nothing but plasma, right? Okay, then next one is, a drug can slowly absorb, may fail to show its therapeutic response as the plasma concentration for desired effect is not achieved. What the sentence means, what it is saying, that is, if the drug will having less solubility or less absorption, it is definitely fail to show its therapeutic response, right? Then on other hand, what is going to happen? A rapidly absorbed, that is, a fastly absorbed drug may attain therapeutic level and to elicit its pharmacological effect. See here, you need to remember two things here. If your drug is having faster absorption, then it is showing good therapeutic response as well as compared with slowly absorbed drug. The name itself indicating that if it is slowly absorbed, then how can you say that it is producing some therapeutic effect at particular level of plasma drug concentration? So we cannot say that. That's why we are not considering here the poorly absorbed drug, right? You got my point? Then that is what both rate as well as extent of absorption is important. Rate, the amount, how much amount is going to absorb, in the extent to extent means what, enough to producing its pharmacological effect. So both are in place an important role to know the absorption of drugs through the GIT. If you understand, once if you understand the concept, then it is very easy to learn coming topics, right? Then how the absorption occurs in the following drugs of administration. I think you already studied the dosage forms. Okay, what are the dosage forms where you need to administer those types of dosage forms, right? So how many routes are there? How many absorption occurs through the following drug administration sites? So how many sites are there for absorbing your drug? That is oral, sublingual, sublingual, where you need to administer this drug under the tongue. Will avoid first pass metabolism because it will be break down in stomach and liver. Your sublingual dosage form avoids first pass metabolism. Okay, so then break down in stomach and liver. And then transdermal dosage forms. Those are, for example, contraceptive. They may have very slow absorption, or else you may take one more example as a transdermal gels or ointments. Okay, they may have very slow absorption because they have to cross so many barriers which is present on the upper layer of the skin, wherever it is. Okay, then third one is rectal. Use when patient is vomiting. Okay, when patient produces some nausea and vomiting conditions, you can use rectal type of administrations. And then intramuscular injection, where you are going to use this, you have to inject this injection into the muscle. Then subcutaneous injection, this is also having slow absorption. For example, insulin, this is a very famous example, and you all know that the patients who are taking insulin, they have to take by subcutaneous route only. I think you know, and you tried in hospitals also. And then my miscellaneous absorption occurs from which routes? Inhalations. Okay, intranasal, the dosage form which you are going to insert it to the nasal route through buccal mucosa. Then eye, nose, ear drops. Okay, these are the exceptional or miscellaneous routes of absorption of drug administration site. If oral route from stomach and intestine to portal circulation. So most of the drugs, most of the dosage forms, in your daily life, you have to use more tablets as well as capsules, right? For treating such respective diseases and all disorders. If you take a tablet, first where it will enters into the stomach, then it will enters into the intestine. It is showing its mechanisms, ADME mechanisms. Okay, so it is one of the route. And then if per rectal, from rectum to systemic circulation, where it happens, the site of administration is rectum and the systemic circulation acts as a plasma. In case of oral, your site of administration from mouth and to circulation into stomach or else or intestine. That is from the site of administration to the systemic circulation. Your drug may not be changing its original form. Your drug will be entered as it is. As it is, original form, it will enter from site of administration to the systemic circulation. Okay, it produces so many desired mechanisms. Okay, then are you clear about this? Yes. If you have any doubts, just contact me, ma. Okay. No need to worry about your biopharmaceutics subject. The concept is important in biopharmaceutics. I am telling you about biopharmaceutics only, not for pharmacokinetics. Pharmacokinetics, you need to practice the reactions and all for 2 to 3 times or more than 5 times. All are having calculations. In case of biopharmaceutics, in that ADME mechanisms, you need to learn about the concepts. First, if you know the concept, then you can write by your own. Okay. Then intramuscular, where it from site of administration, muscle to the systemic circulation. And one more important, very important thing, in case of intravenous dosage forms, those are injected directly through the systemic circulation through veins. It doesn't have any absorption. So no absorption is needed if the dosage is given by intravenous route. So this is about your drug absorption and routes. So coming to the cell membrane, before going to learn what are the mechanisms, what are the transport mechanisms of your drug from site of administration to the systemic circulation, you need to know about the cell membrane. I think you might have studied in anatomy physiology, right, subject. Also called the plasma membrane. The cell membrane also called the plasma membrane, plasma lemma, or phospholipid bilayer, right? The plasma membrane is a flexible yet steady barrier that surrounds and contains the cytoplasm of the cell. So what it contains, your plasma membrane is very flexible, that is, it is easy to break down, and that surrounds and contains the cytoplasm of the cell. Then what the cell membrane mainly consisting of? It is consisting of lipid bilayers as well as proteins. Which type of lipid bilayer? It is phospholipid, cholesterol, glycolipids. These all are of lipid bilayers. And what are proteins? Integral membrane proteins, lipid and core proteins, peripheral proteins. These are the types of proteins, and the above these are the for lipid bilayers. Okay, so the components which consisting of your lipid bilayer, which the lipid bilayer is presenting in your cell membrane, it consisting of phospholipid, cholesterol, glycolipids, right? Okay, then what about proteins? You have three types of proteins: integral membrane proteins, lipid anchored proteins, peripheral proteins. So all of you take your notebooks and pens. You have to write what I am telling. Then you can understand it very easily. You just write the running notes for this, right? I will check it in one class running notes. Don't think that this is an online class. Madam did not check anything and all. Definitely I will do checking. So you have to write it on your notebooks. I will definitely test you if you are writing or not. So this is the structure of cell membrane. See here, ma. See here. This is hydrophilic region. See here, hydrophilic heads and hydrophobic tail part. Okay, these are the transmembrane proteins. And see, on the top of the cell membrane, it is having hydrophilic regions. And the bottom of the cell membrane, it is having hydrophilic regions. So these are called, these are looking like heads. And this green color is looking like tails. So these yellow color hydrophilic regions are easily solubilized most of the drugs, because they are water lovable, right? They are easily soluble. So this hydrophilic head part and hydrophobic tail part is having phospholipid, right? So this pink color is integral proteins. This is the glycoprotein and this is the carbohydrate chain. Here you need, this is the plasma membrane structural component, ma. And here you need to focus it only on hydrophilic regions and hydrophobic regions, and how the cell membrane structure will be. Suppose if you take any dosage form onto the skin, so you have to administer the top layer of the skin, right? You have to spread it on the particular area of the skin. Then it slowly diffuses through this hydrophilic region, then to this tail part, then hydrophilic, then the internal layers of the skin. So likewise, your absorption of drug will be takes place from site of administration, that is skin, to the systemic circulation, that is nothing but plasma. Have you understood the plasma structural components of this diagram? So once you understand this, it is very easy. So how many types of proteins are present? So you have to learn in this pattern. First, you just learn the definition of biopharmaceutics. Then you go for absorption definition. Then you go for cell membrane structure and components. So what it contains exactly, how the cell membrane will be useful for drug absorption. So where, right? So first you have to focus on these topics till now what I discussed. And next, how the movement of substance across the cell membrane. See here, ma, this is a cell membrane. I already said you, this is the head part, this is the head part. These heads are joining through the tails. See here, gases like carbon dioxide, oxygen, these will easily enters to the inner layers of the skin or systemic circulation. Here it is systemic circulation, you can assume it as. Then hydrophobic molecules. These hydrophobic molecules absorption is little bit slow as compared with gases. You have to observe the diagram very clearly, ma. So you just put more concentration on these diagrams, then you can understand the concept, right? Then smaller polar molecules. See here, polar molecules, these are also absorbing easily. And large molecular proteins like glucose and all, the absorption is they doesn't absorb properly. Why? Because these are having large molecular weight, so it is very tough to absorb these large molecular weight. Then how you will absorb, how you will achieve good absorption for these type of drugs? You can modify that change by adding a salt form or by increasing the solubility of particular drug. Then you can achieve it faster absorption. Then charged molecules also. So amino acids and all, the absorption is very less. So here I have explained you 3 to 4 components how the absorption through cell membrane will be. If the drug is not absorbed properly, if you have to adjust the solubility of the drug, or else if you have to change the structural alteration, if you have to change the structural alteration, or if you have to add some salt forms and all, if your drug will be insoluble, right? Are you understand clearly? Okay then. So I'll proceed for next slide. Then coming to this, these are very, very important things. Um, in your examination point of view, they will ask you the mechanisms of drug absorption. Till now, you know the structure of cell membrane, and you know that what is that absorption of GIT and all. And then I would like to explain you about mechanisms of drug absorption. You have 9 to 10 types of mechanisms are there. Majorly, these mechanisms of drug absorption can be classified into 3 types. Transcellular, also called as intracellular. The name itself indicating intracellular, that the mechanism will be happened in intracellular, within the cell. See here, within particular cell, that is called intracellular or transcellular. And second one is paracellular or intercellular, intermediate, same like intermediate means what? Between. So same like that, intercellular. So here 2 cells are there, so the transport of drug absorption will happen in between the cells. See here, paracellular or intercellular. And third one is vesicular or corpuscular transport or endocytosis. See here, ma'am, once again I am reading: vesicular or corpuscular transport, endocytosis. You have to adopt the different terminology. Till now, I think you could not cross all these terminology and all. Right now, you have more idea on hospitality, and then what is that pharmacology subject, histopathology, clinical pathology of the drugs and all diseases, like So you have to adopt the terminology, major thing here. So the three broad categories are transcellular or intercellular transport, paracellular or intercellular transport, vesicular or corpuscular transport, or also called as endocytosis. See here, ma. Transcellular, it happened between two cells. See, sorry, it happened within the cells. See here, this is within the cells. And in case of paracellular, it happened between the cells. And in case of vesicular, it happened within the cells, endocytosis. Mm, coming classes, we will discuss about clearly by individual mechanisms of drug absorption. First, under transcellular or intracellular, we will discuss it now. Transcellular or intracellular transport. It is defined as the passage of drugs across the GI epithelium. There are three steps involved. What are those? Permeation. Permeation is nothing but the diffusivity of your drug molecule, which is presented as an dosage form. If your drug is having some diffusivity to pass through its dosage form and it enters into the systemic circulation, so it should have some permeation for easy crossing of your cell membranes. That is nothing but the word permeation. Permeation of GI epithelial cell membrane. So every cell is having some permeation capacity. Once your drug will having enter through, and drug is entering or crossing through this GI epithelial membrane, it is easily diffused into systemic circulation, right? That is nothing but permeation of GI epithelial cell membrane. And then movement across the intracellular space, that is cytosol. And then permeation of the lateral or basolateral membrane. The various transcellular transport processes are involved in drug absorption are, see here, again, your transcellular or intracellular transport will be divided into two types, right? Passive transport process and active transport process. Under passive transport process, you have four types: passive diffusion, pore transport, ion pair transport, facilitated diffusion. See ma here, transcellular or intercellular, sorry, intracellular, transcellular or intracellular, that is, this absorption mechanism is happening within the cell, intracellular. You have two types in that: passive transport and active transport processes. Under passive transport, again you have four types of mechanisms of drug absorption, that is passive diffusion, pore transport, ion pair transport, facilitated diffusion. Did you write it all? Right. And next, under active transport process, you have two types: primary active transport, secondary active transport. And again, your secondary active transport will divide it into two: symport and antiport. First, we will discuss about passive transport or passive transport processes. Under that, first we will discuss about passive diffusion. Okay, I already discussed about passive diffusion process in previous class, and again I would like to go with this only. Passive transport process, the transport process do not require energy than other than that of molecular motion, that is Brownian motion. What do you mean by Brownian motion? The randomized movement of the drug particles or solid particles. See here ma, the randomized orderless movement of the solid particles or drug particles is nothing but Brownian motion to pass through the lipid bilayer. See ma'am, the transport process do not require energy. It do not require any physical energy other than the particles are having the own Brownian motion to pass through the lipid bilayer. Passive transport process can be further classified into four types. I already told you na: passive diffusion, ion pair transport, pore transport, facilitated diffusion or mediated diffusion. Okay, see ma, here in this graph, they are passive transport mechanism is going to explain here. See diffusion, these are the diffused particles. See ma, your drug particles. So these will be diffused into the systemic circulation. See here. In case of facilitated diffusion, this is also same thing happened, but what is the major difference between normal diffusion and facilitated? It promotes more absorption of drug particles. That's it. That's it. Okay. So passive diffusion is also called as non-ionic diffusion. It is the major process of for absorption of more than 90% of the drugs. That means the 90% of the drugs will follows this passive diffusion for its absorption through GIT. It is defined as the difference in the drug concentration on either side of the membrane, right? Then how you could know this passive diffusion process and all? By knowing the Fick's first law of diffusion. So he is the scientist of Fick. He only explained about this mathematical derivation of passive diffusion. I already told you in the last class. Then how it will be, the which states that the drug molecule diffuse from a region of high concentration to the low concentration. That how much time, where it will get some equilibrium. For that much time, the drug will be transported from high concentration to the low concentration, right ma? Okay, the drug diffused from high concentration to low concentration until it reaches the equilibrium. That is nothing but your passive diffusion. Then dq by dt is the equation. Dq by dt is the rate of diffusion. Rate of diffusion. That is dak m by w by h into cgit minus c. Right? This is the dq by dt is rate of diffusion, d is diffusion coefficient, a is the surface area, km by w is the partition coefficient between the membrane and the bulk of the solution. That is nothing but GIT, right? And then H is the thickness of the membrane, GL lumen, and then concentration of drug in GIT, and then concentration of drug in plasma. I explained you already. If you have any doubts, go through the first class, okay? And then I thought that I can stop my class here because today you learned so many things. If you go on learning means definitely you will get confusion and all. So first you have to focus it on the learning of cell membrane structure, cell membrane composition. First you have to go through that. Then coming to the mechanisms of drug absorption through GIT, how many types are there? Under that, now we are discussing about transcellular or intracellular transport, right? So you have to read all the types and all, and in that you read about passive diffusion and ion transport facilitated. We will discuss it in next class. Okay, from till now I completed these many topics. You have to read it and be ready for next class. Okay, right.